Thanks for joining today. I hope that you and your families are staying healthy in this challenging time. But I know that many scientists right now are working incredibly hard, and we are really proud to be a technology provider to the scientific community. So thanks to everyone for their work. And what I'm going to do today is give you an overview of the Incucyte, which has had a very purposeful journey, as you will see shortly. As research is becoming more complex, both in the terms of questions that scientists are wanting to answer, but also the cell models that are required to provide those answers, new demands are placed on technology. And live cell analysis is a key technology that's evolving quite rapidly to adapt to those demands. So I'm going to give you a brief introduction to the history of the Incucyte and the impact of the Incucyte, and introduce you to the newest member of our portfolio, the Incucyte SX5. We have a bit of a milestone to celebrate, and that is 15 years of the Incucyte. Incucyte was launched in 2005 as an automated cell culture quality control tool. As it still does today, it sat inside a tissue culture incubator, and it imaged flasks around the clock to yield fully automated confluence measurements so that researchers could document and make informed decisions about their cultures. And over time, new features and new applications were introduced, and the Incucyte really transitioned into a very flexible platform, handling a range of vessels, flasks, dishes, and microplates to do live cell imaging and analysis across the entire cell biology workflow, from cell culture to manipulation to assay and analysis. And it supported the workload of an entire lab with a capacity to have multiple ongoing experiments simultaneously. The Incucyte was, and still is, truly enabling access to live cell insights. Then in recent years, we further expanded that accessibility by removing more pain points found in traditional image-based technologies and also launching a more affordable Incucyte. But at the heart of Incucyte product development, it has been and always will be a true applications approach. We start with the question that the researcher is asking and provide all the things that are needed to get to that answer. So whether that's reagents or specialty microplates, and the exact software tools, all of those things we develop and then launch a full application. And the number of applications are expanding every year. This year, we're continuing with that applications approach. We are continuously being asked to help answer more and more questions that are more complex and using more complex cell models. So in response to that need, we're very excited to launch the Incucyte SX5. And we're going to get to the details on that very shortly. But before we get to those details, we want to acknowledge the Incucyte community. This is a global community in 50 countries that's published almost 4,000 peer-reviewed publications. And these publications have been cited over 73,000 times. And just every one of these publications are utilizing live cell analysis. And what we're most excited about is, we see in these publications, researchers are really branching out into exciting areas and making very impactful discoveries with the Incucyte. As you can see here, the number of citations in journals with impact factors of 10 and higher. And this is something we're really proud of the Incucyte community. And now with the Incucyte SX5, the number of questions that can be asked and answered are expanding even further. The SX5 builds on our market-leading design. It keeps cells stationary and undisturbed inside the incubator. Our unique patent-pending optical system offers up to 5 different fluorescence channels, up to 3 at a time in a single experiment, plus HD phase imaging. And we've been very purposeful in selecting these channels. Unlike systems that offer a blue fluorescence channel, for instance, we include a long wavelength near infrared fluorescence channel for reduced phototoxicity, which is exactly what a live cell experiment would need. In addition to the new system, we're including a suite of reagents and software for application support that is truly enabling. Whether you are studying complex immune tumor cell interactions or synaptic activity in neuronal co-cultures, or even metabolism in cancer cells, you can achieve this with a single flexible platform. And that the Incucyte will be able to answer any of the questions for your team and really let the team focus on what are the possibilities instead of having to wrangle technology. So with that, I will turn it over to Dan Appledorn, who will get to the really exciting part, which is showing real-world application data from the Incucyte SX5.
Thanks, Kim. It is really my pleasure and honor to be talking to this audience about our new instrument, the SX5. Just a brief rundown of the agenda. I will first be talking about 3 new 3-color applications that we have developed for the SX5 using the additional color channels. That is how we add additional colors to our immune cell killing application, multiplexing that with our new cell cycle reagent and an apoptotic marker, and then taking a little bit of a step in a different direction and talking about an oncology application and looking at a breast cancer selective drug called CB839 that's selective for triple negative breast cancer cells. And then I'm going to present a new application for us that is launching at the same time as the SX5 that it specifically looks at measurements of intracellular ATP over the course of time using a live cell imaging approach. But before I get started in too much detail with the immune cell killing application, I'd be remiss if I didn't talk about this image that we're seeing here on the right-hand side of the screen. Here we see HT1080 cells that are labeled with our Incucyte reagents in green, orange, and near IR. The green cells obviously appearing green here, the orange cells with a little bit of a red flavor, and the near IR reagents in blue, utilizing the RGB approach to really visualizing cells and able to differentiate them. And with that, I'm going to start this movie so you can see how these cells look over the course of time. As you can see, these Incucyte reagents are restricted to the nucleus of cells, facilitating the accumulation and counting, enumeration of cells over the course of an experimental timeline, now enabling measurements of proliferation of 3 independent cell types within one multiplexed culture system. So jumping into our immune cell killing application. The tagline here at the top, more channels results in more insights and allows users to multiplex our immune cell killing application. Just to simplify this for you, I know it's a busy slide, we have labeled in this case our target cells, which are A549 epithelial cells, with our Incucyte orange reagent. We have labeled our effector cells, which are PBMCs, with our Incucyte Cytolite Rapid Green reagent, and we have also included a new reagent for us, which is annexin near IR, which will be an indicator of apoptotic cell death. And what the images are showing on the top side are cells, target cells in the context of activated PBMCs, activated in the way that's shown here with CD3, CD28 beads. And on the bottom panel are non-activated PBMCs. So these PBMCs are then added to the target cells and measured over the course of time. Within 24 hours, you begin to see rapid proliferation of the PBMC population, as indicated here in green, as well as a decrease in the number of the target cells, which are indicated in red. By 48 hours, it's very obvious that you have an induction of the apoptotic signal in the near IR channel and a significant proliferation of the effector cells and reduction of the target cells. So in a very quick, easy to look at sequence of slides here, you already begin to see the applicability of the 3-color strategy for immune cell killing applications. But as many of you know, the Incucyte isn't about pretty pictures, all about pretty pictures and movies. We quantify our biology using those images over the course of time, which is represented in this slide here. The first graph on the left, the y-axis being the target cell count, and the x-axis in all of these graphs being time. We see that with no PBMCs, we get a dramatic proliferation of the target cells, which is no surprise. Again, these are A549 cells labeled with the Incucyte orange reagent. In the presence of activated PBMCs, you see that there's a dramatic reduction of those target cells. And interestingly, in this culture, we found that even in the presence of non-activated PBMCs, we saw a reduction in the number of target cells within the culture. This is kind of an interesting observation for us, and it a lot of time depends on the donor from which we get the PBMCs. It also depends on the target cells, but an observation nonetheless. In the middle of the graph here, we're showing the proliferation of those PBMCs over time, those effector cell populations. And on the right-hand side, we see the induction of the Innextin-5 signal of the target cells. So again, no PBMC control, we see no cell death. We do see a little bit of cell death when the PBMCs are added. Those are probably dead or dying PBMCs as the culture is initiated, and then a rapid cell death of the target cells as the experiment progresses. This kinetic data, we believe, provides more biological insight. Again, I mentioned PBMCs from different donors have significantly different kinetic profiles. That data is not shown here, but we've seen that in multiple experiments. We also can differentiate the induction of the apoptotic signal relative to the effect on proliferation of both the target and/or the effector cells. Our automated 96-well throughput enables testing of multiple conditions. As I'll show you in future slides, we can test many different effector to target ratios. We can test from several donors. We can test specific activators. This, of course, was a very general activator of T-cell activity. But we can test a whole host of different activators, specific or non-specific, which ultimately results in increased productivity within your lab. And lastly, I'll mention that because we're making repeated measurements over time, we provide a statistical robustness and data confidence that is really unparalleled. Quickly transitioning to how we utilize our Incucyte cell cycle reagent, just a very brief introduction to the reagent first. The schematic at left shows you how the sensor in this case changes as the cell goes through the cell cycle through in G1, an orange phase, through the G1S transition through a yellow phase, and as it moves through the SG2M phase is in green fluorescence. We have qualified this reagent on A549 cells first by exposing them to a thymidine block, washing that block out, and then following those cells over the course of a 72-hour period, in which the cells went through 3 cell cycles. And so you can very clearly see the cells cycling through each phase of the cell cycle over the course of that 72-hour period. We've done this in multiple different cell types. Like I said, those were A549 cells. We're showing here on the right-hand side HeLa cells that are also stably expressing our cell cycle indicator. And just giving you a visual depiction of how that works actually within cells. At 0 hours here, this cell is in G1. And if you follow that cell through 2 hours, 4 hours, you see that that cell transitions through G1 into the S phase where it is fully green by 7 hours and in S phase, through mitosis by 11 hours, and then back to G1 within the 16-hour time point. So this 2-color, we're using 2-color channels here, application allows us to measure cells at which stage they are in the cell cycle, And then by adding the annexin near-IR reagent, we can take advantage of all three channels and make measurements of not only cell cycle and cell proliferation, but also cell death, all in a multiplexed way. So in this experiment, what we did was kind of took a page out of the immune cell killing application, and we took target cells, in this case MDA-MB231 breast cancer cells, and labeled them with a cell cycle reagent. And then exposed them to either activated PBMCs or non-activated PBMCs. And before I talk about the experiment at all, I just kind of wanted to play this movie and let you decide what's going on here. So I'll just quickly play this. We see a slight proliferation of the PBMC population in the movie on the left compared to the movie on the right. By about 30 hours, we start to see an increase in the annexin near-IR signal, which is an indicator of cell death in the left. We see some near-IR signal in the image on the right. We see a dramatic change in the morphology of the cells by 2 days in the image on the left compared to the one on the right. And by the end of this experiment, I'm just going to pause it here. One major observation that we had was not only do we see significant cell death, as noted here in the near-IR signal, but we see an arrest of cells in G1, as indicated by the number of number of orange cells in this image here, compared to the image at right, where we see representation of all different phases of the cell cycle, represented in orange, yellow, and green. So we wanted to get a better understanding of that and quantify that. The first graph I'm showing you here is just a simple graph showing you proliferation of those cells over the course of time. The y-axis being the total cell count. This is the target cells now over the 72-hour period. In the no PBMC control, you see rapid proliferation of these cells. And as we increase the number of PBMCs, this is about a 10-to-1 effector to target cell ratio. We see inhibition of growth, but also induction of cell death. As we observed in the images, the remaining living cells are arrested in G1. To quantify that, the next slide here shows on the y-axis the percentage of cells in G1 on the left-hand side and the percentage of cells in SG2M on the right-hand side over time. As you can see in the no PBMC control, so these are just MDA-MB231 cells going through their normal cell cycle, we see that the level of cells in G1 fluctuates anywhere between 40 and 50%, whereas as you add the increasing numbers of PBMCs to the culture, maxing out at a 10-to-1 effector to target cell ratio, we see an accumulation of those cells that are remaining at the end of the experiment, which we believe are live cells, that 90% of those cells are arrested in G1. Obviously, there's a correlated decrease then of the number of cells that are arrested in SG2 and M, which was a pretty fascinating effect for us, one that we have little understanding of, but one that we'd love to dig in more detail. In addition to making measurements of cell cycle, as I mentioned, we're also making measurements of cell death using the annexin near-IR reagent. On the left-hand side is what we call a microplate graph view of this data over the full course of the experiment. You're seeing every single well within a 96 well and the data that comes from that well. You can see in the top row here, as depicted on the left-hand side, that the density of PBMCs is high, so the highest effector to target cell ratio in the top, and we decrease that twofold as we move down this 96 well plate. On the left-hand side of the plate, adding activated PBMCs, and so you can see in each well the induction of the annexin near-IR response. In the non-activated PBMC controls, as I had mentioned earlier, we do see some induction of apoptosis in non-activated PBMCs, but nowhere near the signal that we see when we have those PBMCs activated. The image is at right, just showing a visual depiction of the quantitative data that I'm showing at left. These are images that are taken at 48 hours. We can then take all that data and graph it on a single XY plane, which I'm showing here. So this is all the same data, just on a single graph with an annexin near-IR signal on the y-axis and time on the x-axis, increasing numbers of PBMCs showing increasing amounts of cell death. So All in all, what we're seeing here is a diminished overall cell count, as well as an induction of a Nectin-5 signal, as well as an arrest of cells in G1. That's shown here, and I kind of put a kinetic spin on this. First, starting at left, we can think about this as over time, what observations can we make? First, we see that the percentage of cells that are arrested in G1 begin at about the 12 to 24 hour time point, whereas relative to the controls, the no PBMC and the non-activated controls. If we move to the right one graph, we see that the total cell count begins to deviate from the controls at around that 24 to 28 hour time point. And interestingly, if we move then further over to the right here, we see that the induction of apoptosis really doesn't start until the 36 to 48 hour time point. So it really shows the power of the Incucyte in making kinetic measurements over the course of an experimental timeline. I thought those movies were so cool that I put them in here twice. I apologize if you feel like this is too redundant, but I really liked how there were multiple different observations that you could make simply by the movies. So we'll just watch them one more time. I have to say the change in morphology here is something that I did not capture quantitatively, but is a fascinating component of this assay as well. And there we go. Okay, so I've talked about immune cell killing, a simple immune cell killing application at first, then one utilizing our new cell cycle sensor. Now I'm going to switch gears a little bit and talk about tumor cells and oncology. So apologize for the rapid change in topics here, but I hope you can follow along. We all know, many of us know, that tumor cells have an increased need, an increased demand for metabolic fuel. Many tumor cells switch to alternative fuel sources, and in this case, we're going to talk about the glutamine to glutamate source for generating ATP. What happens is that glutamine is taken into the cell and exposed to enzymes within the cell, specifically glutaminases such as GLS1, that change glutamine into glutamate. Glutamate can then be used as a fuel to produce ATPs through the TCA cycle. We know through research that GLS1 is critical to mitochondrial metabolism in many different cancer cells, including triple-negative breast cancer cell lines, which will be the focus of the rest of this talk today. There is a drug out there produced by Calithera Biosciences that is in phase 2 clinical studies. It is called telaglenastat, or easily pronounced as CB839. It is a glutaminase inhibitor drug candidate that has been demonstrated in preclinical studies to target tumor cell metabolism. And so what we're going to talk about is how CB839, how the mechanism of how CB839 affects cell cycle and induces cell death. First, by looking at a triple-negative breast cancer cell line, MDA MB231 cells, compared to a receptor-positive breast cancer cell line, AU565. As I mentioned before, the CB839 is known to be a selective inhibitor of triple-negative breast cancer cell lines as opposed to those that are receptor-positive. So what we did is we took our cell cycle label and we expressed it in MDA MB231 cells as well as in AU565 cells, and looked at the effect of CB839 treatment on cell cycle and proliferation as well as cell death. So in the top graph, we're looking at the percentages of cells in G1, and that's depicted in the red lines here. Those cells treated with CB839, you can see they begin to accumulate in G1 or rest in G1 at about the 12-hour time point, whereas the vehicle control continues to cycle through the cell cycle throughout the course of the assay. Interestingly, although the arrest begins at that 12 to 14-hour time point, we only start to see an induction of cell death at around 24 to 28 hours after treatment with CB839. Again, this is a 3-color application using 2 colors using our cell cycle reagent and the other channel using the annexin reagent. Interestingly, and not surprisingly, in AU565, which are receptor positive cells, we do not see any effect on CB839 on arresting cells in G1, nor do we see them induce apoptosis. Just as a control for us, for our own sanity, we also looked at a non-selective inhibitor of cell proliferation or inducer of cell death, specifically carboplatin, which intercalates DNA, causing blockage of the cell cycle. You can see that very easily here. These again are in MDA MB231 cells. On this graph are depicted the percentage of cells in G1, which is represented in red, or in SG2M, which is represented in green. And can see in this case, where we saw an arrest of CB839 in G1, we're seeing arrest of these cells in the SG2M component of the cell cycle. And I apologize for the y-axis here being a little bit different on the top graph versus the bottom graph, but you'll see that it's only at this 72-hour time point do we start to see significant cell death as a component of this experiment. We've done this experiment in the AU565 cells as well as the MDA MB231 cells, and we see a non-selective response. In other words, the effect of carboplatin on both of these cell types is nearly identical, aggregation of cells in the SG2M phase, so a non-selective inhibitor of cell cycle. So now I'm going to transition to a new reagent portfolio for us that has been designed to make measurements of intracellular ATP. And I'm going to do that by continuing the CB839 story. So what we know now is that CB839 has a significant effect on cell cycle, arresting cells in the G1 phase. But now we want to know how that actually happens and what metabolic effect there potentially could be when treating cells with CB839. In order to do that, we have developed a reagent to directly measure ATP production within the cytoplasms of cells. This will provide additional insight into the mechanism by which CB839 can elicit its effect and identify pathways that can be interrogated for new drug targets. A little bit more about our new reagent and new application. This is a novel, non-perturbing reagent that enables direct measurement and visualization of ATP within cells, within tissue culture plates, 96 well plates, or other well plates. These are kinetic measurements that are independent of cell number. So whether you have 5 cells in the well or 100,000 cells in the well, it is independent of the number of cells. Your reading of ATP will be independent of that. It also enables us to, because it is a genetically encoded biosensor, you are able to express that biosensor in one cell type and add it to a co-culture or add it to a mixed culture of cells and be able to make measurements of ATP within that single cell within a complex co-culture system. Where we started with this is very simple. In HeLa cells and HepG2 cells, I'm showing here in the bottom left graphs. On the y-axis is the ATP ratio within these cells, or the amount of ATP relative to the vehicle control within these cells. And what we're seeing is as we block both glycolysis with 2DG as well as the electron transport chain with KCN, obviously the 2 factories for producing ATP, that we can get a concentration-dependent complete reduction of ATP within both HeLa and HepG2 cells, and have done this with multiple cell lines as well. The image I'm showing on the right is a snapshot from our user interface illustrating how we show this within the user interface when we have high levels of ATP or low concentrations of 2DG-KCN. We see brighter fluorescence. When we have lower amounts of ATP with higher amounts of 2DG-KCN, we see lower fluorescent signal. But that's very simple, right? So in this case, it's a very blunt tool. That is taking 2 blockers that block glycolysis and block the TCA cycle. It really doesn't give you a good feeling about how this would work in a more complex model. And so what we did was we applied this to the observations, as I had mentioned before, with CB839. And to make this even, I think, more relevant to in vitro biology, we also did this in both monoculture and co-culture. We know for a fact that stromal cell-mediated drug resistance via nutrient exchange has been documented in the literature. In other words, although we might, with CB839, be preventing glutamate as a fuel for generating ATP, the cell may receive nutrients from cells it's in proximity to in order to gain more fuel for generating ATPs. Cells just have a habit of doing that, especially cancer cells. So one of the advantages, as I mentioned, of the genetically encoded ATP sensor is that you can observe these metabolic changes occurring in cancer cells in the context of co-culture models. So let me get to the data here. So here we have HCC38 cells, which are triple negative breast cancer cell lines. As I showed you before, in triple negative breast cancer cell lines, we see an effect of CB839 on their proliferation and induction of cell death. And in monoculture, that's what you're seeing here. As you increase the amount of CB839, you see a decrease in the amount of ATP. So we are blocking glutamate as a fuel source for generating ATP in these triple negative breast cancer cell lines in monoculture. Well, look what happens when you put them in co-culture. So if you put them next to CCD 1086 SK cells, the 1086 SK cells are now providing nutrient exchange, we think, to the HCC38 cells, providing an additional fuel source for the generation of ATP, and you lose the effect of CB839. This is specifically relevant to the thought of using CB839 as a single treatment in a cancer patient where you know that their cells are in contact to stromal cells. Interestingly, we did find in MCF7s, which are receptor positive, that there was a very transient but very significant reduction in ATP in the presence of CB839, peaking at about 10 hours, but then recovering back to normal levels within about 48 hours after assay initiation. This is the only receptor positive cell line where we saw CB839 have any type of effect. And we would have missed it had we not looked at the full kinetic approach. If we had just taken a 72-hour time point, we would have not seen this kinetic curve that we're seeing here. Interestingly, whether or not those cells were in monoculture or co-culture, we got the same response. And we didn't stop just with the 1 or 2 triple negative breast cancer cell lines or receptor positive cell lines that I'm showing here. We actually extended this panel to 4 triple negative breast cancer cell lines and experimented with those in both monoculture and co-culture. In co-culture, we found that there was very little or no effect of CB839 on ATP levels within triple negative breast cancer lines. Whereas in monoculture, we saw a very consistent pharmacological effect on ATP levels when cells were cultured in monoculture. With receptor positive cell lines, again, we looked at a panel of 4 receptor positive cell lines. There was only one culture that responded to CB839, and that was MCF7s. And so what we're showing here is the pharmacology at that 15-hour time point. The rest of these time points here, we have the full kinetic curves. We saw 0 response. We're just showing here the 72-hour data. But we saw absolutely no response to CB839 in any receptor positive cell lines aside from the MCF7, the transient response that we found in MCF7 cells, thereby confirming the effect of, number 1, the selective effect of CB839 in triple negative breast cancer cell lines versus receptor positive cell lines, and the effect of minimizing the effect of CB839 when those cells are placed in more complex co-culture models. Brief summary. What I hope I've shown here is that SX5 enables more colors, more data, more multiplexing, and most importantly, more insight. I gave you 3 examples of how we've utilized 3 color channels in immune cell killing, with immune cell killing with cell cycle reagents, and the selective effect of CB839 and non-selective cell cycle arrest of breast cancer cells with carboplatin. And then it's very exciting for us, introducing a new application with new reagents to make live cell measurements of intracellular ATP, which enable non-perturbing direct measurements of ATP in live cells over time in advanced multicellular models and independent of cell number. Thank you very much.
All right. Well, I would like to thank the organizers for inviting me to give this talk today. I'm Ted Hinchcliffe, and I would like to acknowledge my postdoc, Charlie Day, who carried out all of the Incucyte experiments that I'm going to share with you today. So I'd like to talk to you about how we use live cell imaging to study cell proliferation in the context of both basic cell biology and in preclinical studies. And I'd like to share with you how we've introduced the Incucyte system into our existing workflow. So here's a quick overview of my talk. I'm going to briefly discuss various cell fates when we're talking about cell proliferation. I'm going to talk about methods that we and others have used to measure cell proliferation over time. I'm going to give you the briefest of histories of how we've used live cell microscopy to study these questions over the last 25 years. And then I'm going to take you through some current approaches, specifically using the Incucyte instrument, and take you through a basic experimental design that we've used from start to finish to show you how we've integrated this into our workflow. So it's important to consider different cell fates when you're interpreting cell proliferation data. So when we look at a growth curve, we're really trying to flatten that curve if we're treating it with a drug or a genetic manipulation. And we have to ask, why is that curve being flattened? So at a very basic level, cells divide, cells die, or they just sit there. And division and proliferation is fairly straightforward. They're going through rounds of cell division, followed by cell cycle, and then division. Those cells may decide that it's time for them to die. This can be a regulated process, and I've listed a couple of those on my talk: apoptosis, autophagy, etc. They can also be cytotoxic. The cells are simply responding to an environment where it's going to cause them to die. And then cells can just sit there. Cells senesce, they quiesce, they become terminally differentiated, and they just stop proliferating. That may be dependent upon a particular mechanism. Cells also will arrest in their cell cycle. If we manipulate them with a drug or we manipulate them genetically, we may see them arrest in a particular area of the cell cycle. And so it's important when we look at these growth curves, we want to think about, why are they not growing the way we thought they would be? There's a variety of different tools that we use to measure and study cell proliferation. Historically, we've looked at the accumulation of a particular component, DNA content by flow cytometry, uptake of a marker like bromodeoxyuridine to look at DNA replication. We may look at a percentage of cells within a population that are expressing a growth marker like PCNA. We'll look at changes in fluorescent markers like we saw in the last talk with the dual color cell cycle marker that gives us an indication of which cell cycle phase a particular cell would be in. There are the endpoint assays that we're all familiar with: clonogenic assays, viability assays. How long has a cell grown into a particular colony, and can we measure that? And then finally, what we have been using in my laboratory for quite some time, which is live cell imaging. This is a continuous measurement method that allows us to watch cells as they grow, stop growing, die, and we can do this rather directly. Here's some examples of what that type of data looks like. For the most part, it's been static data, looking at a proliferation marker within a tissue. You might look at colony formation using crystal violet. Even flow cytometry gives you an indication of it at a particular time point. I do want to point out that something like flow cytometry can be used to give you some dynamic data. Here we have DNA content in drug-arrested cells. And then when we've washed those cells out and over time, we can watch them as they progress into different phases of the cell cycle, looking at their DNA content. But it really doesn't give us the dynamic information that we would like to get from live cell imaging. So let me briefly take you through some of the types of live cell imaging that we do in my laboratory. 25 years ago, this was a very basic process when I was a postdoctoral fellow. These are examples of some of those experiments These are sea urchin zygotes going through their first round of division. This data was taken with a 35mm camera and a timer. And I would sit there in front of the microscope and at various times take pictures using polarization optics. We could do this to look at various drug treatments in the top set of panels. We could also do genetic manipulations by microinjecting a particular protein, in this case a mutant cyclin protein. And we combined these fairly rudimentary experiments with a sort of same cell approach where we would follow an individual cell to an endpoint. That cell would be fixed and then either labeled with a fluorescent marker, or in the case of the bottom set of panels, you're looking at same cell live and electron microscopy, where these cells were embedded, serial sectioned, and the electron microscopy of the individual poles in the multipolar mitotic spindle we used to identify the number of centrioles that were there. As time went on, we switched to doing video microscopy, where we could continually follow these things with at first a video camera and then a digital camera. This is phase contrast microscopy showing two individual cells growing. These are mammalian cells over a period of 50 some odd hours, going through multiple division cycles. We can visualize that. We see the timing between cycles of mitosis and then growth. This is also a very powerful technique that we've used for a long time. One component of this that's important to point out is that not only do we have to be concerned with the microscopy, but we also have to think about cell physiology. So we had to build an environmental system to house our microscope in to allow these cells to continue to grow. These can be purchased commercially. In our lab, we actually built it ourselves. And we've extended this to looking at live cell imaging using fluorescence microscopy. Here's just an example of an individual cell that's in mitosis that is yellow, and it has a chromosome that has not aligned, which is purple. We're looking at fluorescent histone proteins. As the cell goes through division and forms two daughter cells, which are the two yellow circles, there is a micronucleus that forms, which is the little purple dot denoted by the arrow. And this cell will then arrest over the period that would encompass 2 or 3 complete cell cycles. We can watch neighboring cells as they continue to divide in these movies. So those are the red and orange cells that you see, allowing us to see that the conditions are permissive for proliferation, but our particular cell is not able to do that. So there's a lot of strengths to this approach. We've used this for a long time. We think it's very powerful. The single cell resolution in both space and time we think is very important. And it gives us a mechanistic understanding of how various drugs and various genetic manipulations function on a particular cell treatment. But there are limitations to this approach. It's slow. This is a fairly low throughput system. There is a heavy burden on person hours for doing the analysis. When we have moved into a more preclinical translational work where we need to examine multiple cell types, we need to generate isogenic cell clones from an individual cell line using CRISPR, and we need to do combination drug treatments, we find that this low throughput is really a burden for us. There are ways to modify existing microscopes to make them a more high throughput, but the problem with that is that we're now taking one of our core technologies and we're really changing it for something that it wasn't designed to do. And so it was recognizing this that we decided to use a new system, which is an integrated imaging and analysis system. In this case, we use the Incucyte machine that you've heard about already today. And I'm going to talk to you a little bit about how we've used that. Very simply, how we came to use the Incucyte was word of mouth from other scientists. In particular, there was a seminar from a group at the Mayo Clinic, had come over to our institute, presented Incucyte data that was really compelling to us, that this seemed to be something that would fit with our workflow. So in the last few slides, I'd like to take you through just a sense of how we have done our experimental design from really having never used the instrument all the way to being able to take very meaningful data. And I'm going to show you growth curves rather than showing you the cells, because I think in the interest of time, it will really help you get a sense of how we've done this. The first step that we need to do is to figure out what the initial cell density for an experiment is. For different cell types and different growth rates, it's important that we begin with the right cell number. And I think you can see here, in a period of what turns out to be about a week, 160, 70 hours, we're able to determine on a pretty broad scale what is an optimal density for this particular cell type. The same is true for optimizing our drug concentrations. So we have a drug that's got maybe a published IC50 or one that we've determined enzymatically, but we really want to see what's optimal for this particular cell type. And so very rapidly, again, within a week's time, we can generate data that tells us what a good concentration, at least to start our experiments. I also want to point out, if you look at the ends by each one of these graphs, in an individual run, we're able to run multiple wells with the same treatment. That gives us some idea of error. It also allows us to recognize sometimes we'll have a particular well that nothing happened. Is that because the drug didn't work, or is that because maybe we forgot to add it or something else? By doing multiple replicates at the same time, we're able to very quickly get a sense of reproducibility. We can include then multiple runs over multiple weeks. That's a very powerful tool in doing these types of experiments. Once we've determined an optimal drug concentration, we can then test it between two different cell types. We have normal diploid cells in the top two panels and then a set of cancer cells in the bottom two panels. We can really look at how that curve flattening differs between different cell types at similar drug concentrations. And this all comes together when we're looking at multivariable treatment. In this case, we're looking at a drug, at radiation therapy, and then the combination of the two. In this experiment, we're really looking to see whether we get some type of radiosensitizing effect from the treatment of this particular drug. These type of cancer cells, radiation therapy is considered the standard of care, and so it's important to see how the drug interacts with radiation therapy, comparing normal cells with our mutant cancer cells. And so in a single run, we can get a sense of very reproducibly, how does this drug affect this? And I think if you look over in the mutant cell growth curves, you look at the purple line, I think it's fairly convincing that there is a radiosensitizing effect for this particular drug. Another example of a different type of drug used in these studies, where we have, with the yellow line, cancer cells that have been treated with a certain amount of drug. There's a flattening of that curve, suggesting to us that maybe there's a senescence that's being introduced by this particular drug. We can very quickly see, does that occur in normal cells versus our cancer cells, and is there an effect by including radiation therapy, where we see that yellow curve and there's a definite loss of proliferation in response to having radiation treatment as well. This took us a very short period of time from initial seeding cell studies to being able to graphically see how drugs and radiation affect our particular cell type. And the last data slide I'll show you would talk about doing CRISPR modifications of cells, either introducing a known mutation into a cell line or reverting a known mutation back to wild type. The question between these isogenic cell pairs is, does that mutation confer some sort of detrimental cell growth? You can certainly look at plates and get an eyeball idea of whether they're growing or not, but within a couple of days, using the Incucyte, we're able to confirm, yes, this changing this particular mutation doesn't have a dramatic effect on its ability to continue to grow, and that makes it a good candidate for us to be able to study. So as I come to the end of the talk, I want to talk a little bit about how we can use assays that are built into the Incucyte system to begin to ask sort of the why. Do they divide? Do they die? Do they just sit there? There are a variety of existing assays, things like looking at number of nuclei for nuclear counts, apoptosis assays, and cytotoxicity assays that we can use to answer that question. If there is a flattening of the curve, why did that curve flatten? We're particularly excited about the new cell cycle reagents, the dual color cell cycle reagents that were mentioned in the last talk, and combining those with immune cell killing, particularly with the new instrument that Sartorius is bringing out, the SX5, and their ability to do triple fluorescent labeling. We're always looking to get from these population studies back to our single cell microscope, but the more information we can get from a population very rapidly, that makes it easier for us to be able to go back to that single cell scope. And finally, I'd like to conclude with how we feel like this instrument is really added into our live cell workflow. It allows us to do rapid analysis, particularly of multiple cell types, multiple changes to those with drug combinations, CRISPR mutations, and particularly when we have to do combination between multiple variables. We think this is a really powerful instrument for doing that. We like to do a multi-approach. We start with single cell that gets us to a mechanism. We then go to this population instrument that allows us to look at a large number of cells and say, here's the drug, here's the pathway. Initially, we would bring this back to the single cell level to continue to refine our model and make new predictions. But I think with advances in the instrument, just like advances in standard fluorescence microscopy, we're now seeing that we go back to the population again, where we can look at things like cell cycle regulation. Is our drug arresting cells in G1, S phase? Is it a mitotic arrestor? So we see that as a multiple approach going back and forth between these instruments. And we see this as part of a complementary approach. We don't want to try to do one thing. We try to see it in a variety of different ways. We continue to do flow cytometry and clonogenic assays, our single cell microscopy, which is really our core technique in the laboratory, and this integrated microscopy and analysis system with Incucyte. We think these all complement each other. In particular, the idea that clonogenic assays, which are the industry standard for radiation therapy, we feel like data that comes out of our Incucyte experiments really provides a strong complement to that clonogenic assay, and we've actually heard from reviewers that they felt that both of those approaches together really strengthened what it was we were doing. So I think in the interest of time, I'm going to stop here. I'd like to thank you all for your attention, and I want everyone to be well, and I think we're about time to start with some questions. So thanks, everyone, and we'll talk with you after the conclusion of these talks.